Hi, my name is Wolfgang Hankeln and I'm sitting here with Rob Robbins from the US. And you are also visiting us as well here for the meeting. You are mainly here for another meeting, but luckily you found the time to drop by. And first of all, how was your trip?
Well, as Volker mentioned, 17, 18 hours of travel is tiring, but wasn't too bad. This is the first time I'd come through Munich, first time I'd been to the Munich airport. It's a decent facility. And I left Sunday. I spent yesterday being jet lagged, so I'm not so bad today.
And is it your first time here in Bremen?
No, I was here for the GSC meeting last fall.
But this didn't take place here at the Jakobs campus?
First time here. That GSC meeting was at the Max Planck Institute.
And what is your impression about the campus and the surroundings? Do you like it? Or?
Oh, I enjoy it. I mean, part of the reason I'm here in Bremen right now is because I enjoyed the trip the last time so much that I thought I wanted to touch base with Renzo Kottmann and possibly with Frank Oliver Glöckner when I came through. And I was going to this meeting in Copenhagen next week. I sent Renzo a note. He let me know there was this other workshop going on. I looked at the agenda and said, wow, this could be useful for me to see. And the reason it's useful for me is the project I'm working on, this research coordinating network for GSC, this National Science Foundation funded activity in the US to bring the Genomic Standards Consortium work to a broader audience, primarily in biodiversity and ecology. I've been functioning at kind of a high level, sort of almost a theoretical biologist who's all excited about this, the fact that half the world's biodiversity is now visible for the first time and that sort of thing. But the actual hands-on of producing microbial community sequences, I've not been doing that. That's not been something I've been involved in. So when I saw that the workshop was going to be practical hands-on training, I thought, well, I don't want to crash the party and take up a workstation, but if I just come watch it, I will come away knowing more about the details. And with any methodology, there's theoretically what it does, and then there's the practical issues of what actually is hard to make work. Where do the artifacts creep in, and that sort of thing. And so I thought it would be useful to come take a look at that, and that's why I'm here.
So what are you more interested in? Are you more interested in the theoretical part of the short course or the practical part? Or simply meeting people?
Well, actually, I'm interested in all of them. My personal background is I was trained as a biologist years ago. I served on the faculty of a large Midwestern university in the United States, taught introductory biology on television to sometimes 1,000 students at a crack. I think over the years I had more than 10,000 students go through classes I was teaching. So when you've done that, you have a tendency to pay attention to teaching, to pedagogy. It's something you're interested in. Is this being done well? Hard to resist saying, here's the way I would do it, when you watch it happen. Also, one of the things I did after I got out of teaching, I went to the National Science Foundation as program officer for the first time we created a program to fund biological databases. That was when I was at NSF. From there I went to Johns Hopkins and I was the informatics core director for the genome database, which was part of the Human Genome Project. And that sometimes involved coming to Europe to offer short courses on how to use GDB. My first introduction to that was I wasn't supposed to have anything to do with the training. I'd only been associated with the program for a couple of months. My senior scientific colleague there, Peter Pearson, was supposed to provide the training at Heidelberg at, I forget whether it was at Embl Outstation then when it was there or whether it was at the DKFZ, one or the other. But Peter was British. We had a meeting in England. We went to that. He was supposed to take another week and go to Germany and teach this short course, and his mother fell ill. So he hands me the instructional tapes and says, go teach the course. So I've had that experience a while ago of trying to teach complex technology in an international setting where sometimes if you have a language facility issue between the teacher and the student, small issues can become part of the problem, not just a big conceptual one. So I'm interested in the general pedagogy. I'm interested in the details of how it goes. I'm interested in learning some of the things that I don't know myself, and I'm finding that this what I'm going to be doing next week is giving a keynote address at GBIF, the Global Biodiversity Informatics Facility in Copenhagen. They've got a planning meeting going on, Global Biodiversity Informatics Congress or something like that, GBIF, to sort of set a vision for future biodiversity efforts by GBIF. And the message I'm going to be taking to them is that metagenomics and microbial community sequencing, the thing this workshop's all about, are showing us that we just didn't have a clue about what half the world's biomass and most of its biodiversity has been doing. Mm-hmm. And that any effort to try to manage and share and explain all biodiversity information that leaves this out is hopelessly inadequate. So in fact, I just mentioned I saw a manuscript that was in press and marked not for release, and I was checking with Frank Oliver about that, saying, can I at least mention this at the meeting next week, because this is a review of the kind of thing that these people really need.
So based on your experience, how, from a pedagogical point of view, must the course be designed to be successful, a course like this?
Well, what I've learned in teaching is that it's easy for people to learn things that they already have a question for, where what you're teaching them is the answer. If you really want to know how to change out the starter motor in a 1968 Volkswagen van, I can tell you how to do it, and you'll learn it in a twinkling. If I start telling you how to do something else that's equally complex you have no interest in, I'll lose you in a very short period of time. So the idea of trying to establish the intellectual context in which the explanations are going to happen, I think, is important. That probably took place here last week, and I wasn't here last week. But I think that's a really important basic sort of thing, as is assessing what do the students know. You don't want to talk over them, and you don't want to talk under them. And if you've got a mixed class, you have to figure out a way to move around in your level of presentation so you bring everybody to pretty much the same level of understanding. And then you establish the thing, and then you say, what are the details? What do you have to understand to make it work? And then what do you have to understand to actually do it? I think the comment that Volker Meyer just made about incorporating a hands-on experience with students' own data. And he was talking about that in the context of it's always valuable for people to have an opportunity to analyze and explore and look at their own data. I would look at that from the purely pedagogical point of view as if you have data to analyze, you have questions to answer. The other issue is this stuff is complex. My original background is in animal behavior. A steep learning curve means it's easy.
Okay.
What a learning curve is it's a percent of successes out of a set over time.
Okay.
So if you go from 0 to 100% in a short period of time, that's a steep learning curve. It's easy. The problem with a lot of technologies is the learning curve is neither steep nor shallow. It's curved, which means there's some basic information that until you get that, you can't begin to get success. And so to me, the critical thing, if these students are coming to this workshop and it's their first informatics workshop of this kind, is get them past the bottom of that S-curve. Okay. Get them to the point where now they can begin to explore and have some success on their own. Yep. Then it gets hard again at the top when you have to start understanding not just how do I put data in, how do I put specimens in, how do I get results out, but how do I interpret them meaningfully given the limitations of the parameters that I've set, the potential for artifacts in my data, and that's the difference between being the facile early user and the expert who manages the other part of the curve. And I believe teaching has to be tailored to the difficult part at the bottom where you just can't figure out barely how to turn it on. And then this rapid learning phase where you go, I can do this, I can do that, I can do something else, and then, but how do I understand it? That's when it gets tricky.
So based on the people you have met, I mean, you've been here only a short time, right? But the participants you met and also the speakers, what is your impression? Is this the right combination, and were the right people selected to manage what you've just described?
It certainly looks good to me. The key thing to having a successful teaching experience, well, there's two key things. One is to have teachers that have a clue, and the other is to have students who have an interest. And it looks like both those conditions are being met here. The instructors are knowledgeable folks with regard to the software that's being dealt with. The environment is nice, nice setup, workstations for everybody. That's good. The lecture room is large enough relative to the number of students that we're not just all wedged in, sort of thing like that. That's good. So I think this is a good workshop. And I think 2 weeks is about right. I think that if you start getting into 3 and 4 weeks or something like that in a workshop, when you learn things, there's this, I hear it, and I think I understand it. I hear it, I think I understand it, I use it, I consolidate it, and then I begin to build on it. And if you keep getting new stuff over 3 and 4 weeks straight without the consolidation, it's very easy at the end of the 4 weeks to have forgotten the first 2. So I think 2 is about right in this context. The idea of whether or not to bring people back again, I think that whether it's bring people, the same group, back to another workshop, whether it's to make a series of workshops or some other way of giving people who have learned something here at this workshop or at a similar workshop, and then they take it home back to their base of operations, their lab, try to use it, maybe do use it, but then to get some feedback from somebody who's substantially more experienced with it to say, but did you think about? Or you think you have this result, but given the design and the way you used it, there's a potential for an artifact here, and you need to add this control or add this reanalysis or add this secondary sampling on this thing to see if that's what's going on. That's what turns people from being a competent beginner into a beginning expert.
Well, so maybe one final last question. So in general, why do you think it's now the time to make these kind of meetings for bioinformatics? And
well, one of the other things I've been doing is there's an umbrella organization in the U.S. called the Federation of American Societies for Experimental Biology, FASEB. And like any organization, they've got lots of committees, and one of the committees is on data issues. And we respond to government requests for comments and maybe throw our own ideas in the hat. And recently at NIH, a working group came out with some recommendations on informatics, and one of them was the need to increase the workforce through training. And the FASEB staffer with whom the committee interacts a lot sent me a note saying, have you seen this? It was actually just yesterday or the day before. Have you seen this? What do you think about it? And my response was, well, talk about the importance of building the workforce, of improving the quality of workforce, and the expertise has been part of every committee's recommendation on informatics for the last 3 decades. So now is the time, then was the time, tomorrow will be the time. It's always the time for this because we've gone from a period 30 years ago when informatics and computational use was of use to some biologists, where today it's, if not integrally involved in their research, it is close to the edges. And within 10 years, we're going to get to a situation where a person who claims to be a competent biologist but really doesn't understand computation is actually confessing a fairly big problem in their biological education, that's just going to become part of it, because sequence data are becoming so ubiquitous. The increase in sequence data is stunning. We're going to have a whole new set of computational problems that are technical related to that, because let me just throw you an interesting statistic. 20 years ago, 1990, given what network speeds were on typical campuses and how much sequence you could produce for $1, you could spend $1 million producing DNA sequence data and then take your data set and move it over the campus network in 1 second, because it was $5 a base, and so or even $1 a base, $1 million didn't get you a lot of sequence. Today, even though networks are a good 100 times faster than they were 20 years ago, it takes about 3 hours to move $1 million worth of sequence over a campus network. And if the next decade shows the same increase in sequencing efficiency than we've seen for the last 20, by 2020, it will take 30 years to move $1 million worth of sequence data. So the world we're heading into is just this stunning avalanche of sequence data, which no human being on Earth, I don't care how smart they are, can look at piles and piles and piles of A's, T's, C's, and G's and do anything with them without a computer. And so and because sequence data is relevant, not necessarily critically essential, but relevant to every aspect of biology and medicine. Every one of them has some area where understanding can be facilitated through access to appropriate sequence data. So it's close to ubiquitous now. It will be ubiquitous within 10 years, can only be understood computationally. And one of the things that anybody who's worked with computers a little bit knows is that they're really good at doing a lot of things really fast, including a lot of wrong things really fast. So one of the ways to really have a major career, not necessarily career ending, but certainly career adjusting event is to misunderstand your computational algorithms, run a lot of data through, think you found a great insight, get the publication out, the reviewers don't spot the bug either, and then it's found out that what you discovered is an artifact. that's problematic. Nobody wants to do that, and that's why adequate computational training is going to be critical. And then it's also going to be critical on the positive side, because buried in this avalanche of data that are going to be coming our way are going to be, especially in the field of microbial ecology, some very substantial, and by that I mean as large as anything we've seen in the last 100 years, of new insights into the way life works on this planet. And it's going to come from, some of it is going to come from some young scientist who brings new eyes and a new way of thinking about it and computational tools, and looks at the information and says, I think I see this pattern. And the reason it's going to, some of these key insights are going to come from young scientists or new scientists. I mean, you could bring a physicist in or somebody from another field, but someone who hasn't spent decades thinking about how it is, but it just goes in and looks with sort of open eyes. Because what you see when you're a scientist, what you see depends on the light source you're using, meaning what's your technique, what does your methodology allow you to see. It depends on your background, and it depends on where you're looking, and it depends in part on your personal biases. In primatology, for example, when the first primatologists were all male, they went out, studied chimpanzees and rhesus monkeys and baboons and whatever, and they saw all these very complicated male dominance hierarchies. They didn't see anything going on amongst the females. And it wasn't until female primatologists went out and started doing research, they discovered a lot of complexity in the female behavior too. The bias that all scientists bring, that's going to take some work to get over the understanding microbial ecology, is we are in fact large-scale multicellular organisms that our biology is heavily shaped and constrained by that multicellularity. And so we're so used to thinking about the way multicellular organisms function, where there's an individual with a boundary and an inside and an outside and all that kind of stuff, and that metabolic pathways happen inside an organism, that it's very hard to get your head around a microbial environment where there are millions of cells not physically attached to each other, where the metabolic pathways happen in that overall milieu, not necessarily within an individual cell. It's really a different way of thinking about things, and the more fresh eyes we can bring to bear on that, sooner we'll get someplace.
Well, thanks a lot for sharing all that knowledge with us and answering my questions patiently.
Sure.
You answered most of them without me having to ask them. So
well,
that was very...
well, that's one of the advantages of having me sit in the background for the other interview. I just memorize the questions and let rip with the answers. You can skip the script or whatever. So, thanks for having me.
Thanks a lot.
Okay.